Good morning, everyone. I'm delighted to welcome you all today. Thank you for joining this session. Today, we will be discussing a very important aspect of pharmaceutical development, the stability indicating testing method from a regulatory perspective. On this slide, you can see the title of our presentation, led by Dr. S.K. Singh, Ph.D. Over the course of this session, we will explore the key principles behind stability testing, regulatory requirements, and best practices for developing reliable stability indicating methods. Let's get started and delve into this essential topic together. Let's talk about stability testing and why it's so important. Essentially, stability testing helps us understand how the quality of a drug might change over time. This is crucial because we need to know how long a product remains effective and safe for use. When we perform stability testing, we evaluate key factors that can impact the drug's integrity. These include temperature, humidity, and light. Each of these elements can influence the chemical stability, physical appearance, or even the potency of the medication. By assessing these factors, we can determine the appropriate storage conditions to maintain the drug's quality. This process also helps us establish the shelf life, or how long the drug remains suitable for use before it begins to degrade. Furthermore, stability testing provides the regulatory foundation for our product's storage guidelines. For example, we follow the ICH-Q1 guidelines, which set international standards for stability testing procedures. In essence, stability testing is a key part of ensuring that our medicines are both safe and effective from production until the point of use. It informs our decisions on shelf life and storage, giving confidence to both manufacturers and consumers alike. Let's take a closer look at the regulatory framework that guides our stability testing and analytical procedures. First, we have ICH-Q1, which outlines the stability testing requirements we need to follow to ensure our drug substances and products remain safe and effective over their shelf life. Then, there's ICH-Q2, R2, which focuses on the validation of our analytical methods, making sure they are reliable and reproducible. Moving on, ICH-Q14 is all about the development of analytical procedures, emphasizing the importance of establishing robust methods from the start. Lastly, ICH-Q6A and Q6B set the standards for specifications, helping us define acceptable quality criteria for both drug substances and drug products. Now, a key concept here is the stability indicating method, or SIM. This is an essential analytical method because it accurately measures the active pharmaceutical ingredient, or API, in the presence of any degradation products. Essentially, a SIM must be able to separate those degradation products from the API to give us a clear picture of the product's stability. For a method to be considered a true SIM, it needs to be specific, meaning it accurately identifies the API without interference, and sensitive enough to detect even small amounts of degradation. It also has to be validated, ensuring that it performs consistently under various conditions. Ultimately, the goal of a SIM is to demonstrate the product's stability profile, giving us confidence that the drug will perform as intended throughout its shelf life. Connecting this to our overall theme, understanding and implementing these regulatory standards and stability indicating methods are fundamental to developing safe, effective, and high-quality pharmaceutical products. Now, let's take a closer look at the different types of stability studies we conduct. First, we have long-term studies, which are essential for understanding how a drug product behaves over its intended shelf life under normal storage conditions. Then, there are accelerated studies, designed to predict long-term stability by testing the product under elevated stress conditions to see how it might degrade faster. Intermediate studies fall somewhere in between, often used to support stability during the product's development phase. Next, we have stress, or forced degradation, studies. These are intentionally designed to challenge the stability of the drug by subjecting it to extreme conditions like heat, light, or pH changes. They help us understand the degradation pathways and identify potential impurities. In-use stability studies evaluate how the drug performs during typical use, including factors like storage after opening. Photostability studies, on the other hand, assess how light exposure affects the product's stability, which is crucial for products that might be exposed to light during storage or use. Moving on, the purpose of stability indicating methods is fundamental to our work. These methods allow us to detect degradation products, ensuring we can monitor any changes in the drug's quality over time. They are key to confirming that the drug remains safe and effective throughout its shelf life. These methods support critical activities like assigning shelf life, preparing regulatory submissions, and continuously monitoring critical quality attributes, or CQAs, that are vital for product consistency. Altogether, these stability studies and methods help us safeguard the quality of our pharmaceutical products, ensuring they deliver the intended therapeutic benefits safely to patients. Let's talk about forced degradation studies, which are really key for SIM. These studies are conducted under extreme conditions to simulate potential degradation pathways of the substance. We typically use acid or base hydrolysis to see how the compound reacts in different pH environments. Oxidation is another critical condition, helping us understand how the substance withstands oxidative stress. Thermal degradation involves exposing the sample to high temperatures to observe its stability under heat. Lastly, photolysis involves exposing the compound to light, which can cause breakdown or changes. The main purpose of these studies is to identify degradation pathways, so we can understand how and where the molecule might break down. It also helps us prove the method's specificity by showing that our analytical procedure can distinguish the intact drug from its degraded forms. Overall, these studies are crucial because they ensure the robustness and reliability of our analytical methods, giving us confidence in the stability and quality of the product throughout its shelf life. Now, let's take a look at the analytical techniques we commonly use in our stability testing. One of the most prevalent methods is HPLC, or high-performance liquid chromatography, along with UPLC, which is a more advanced and faster version. These techniques are essential for separating and analyzing complex mixtures. For volatile compounds, gas chromatography, or GC, is the go-to technique because of its effectiveness in analyzing substances that easily vaporize. When it comes to impurity profiling, LCMS, or liquid chromatography mass spectrometry, offers high sensitivity and specificity, helping us detect even trace impurities. We also utilize UV spectroscopy, which is a quick and non-destructive method for identifying and quantifying compounds based on their light absorption. Additionally, stability chambers are critical for testing how products hold up under various environmental conditions, simulating real-world storage scenarios. Moving on to what makes a stability indicating method robust and reliable, first and foremost, specificity is crucial. It ensures the method can accurately distinguish the active ingredient from its impurities and degradation products. Accuracy and precision are equally important, as they confirm that our measurements are correct and reproducible. Linearity is about ensuring our method provides proportional responses across a range of concentrations, which is vital for quantification. Robustness tests how well the method withstands small variations in experimental conditions, maintaining consistent results. Lastly, detection and quantitation limits define the smallest amount of analyte that can be reliably detected or measured, which is fundamental for sensitive analysis. Altogether, these techniques and characteristics form the backbone of our stability testing, ensuring that our products meet quality standards throughout their shelf life. Let's talk about specificity, which is a core requirement in our analytical methods. This means our method must be able to distinguish the target API from other substances present in the sample. Specifically, it needs to separate the API from impurities and degradation products effectively. It's crucial that our analysis shows no interference from these other components, ensuring the accuracy of our results. To demonstrate this, we rely on stress studies, where we intentionally degrade the API under different conditions to see if our method can still accurately identify and quantify it. Additionally, Peak purity analysis helps verify that the peaks we observe truly represent the API without overlaps from impurities or degradation products. Overall, these steps confirm that our method is specific enough to provide reliable and precise results, which is fundamental for ensuring the quality and safety of the pharmaceutical product. Now, let's take a closer look at the validation parameters outlined in ICH Q2R2. These are essential for ensuring our analytical methods are reliable and fit for purpose. First, we have specificity, which is about the method's ability to measure the analyte accurately without interference from other components. Next is linearity, confirming that our method produces results proportional to the concentration across a specified range. Then we have accuracy, which tells us how close our measurements are to the true value, and precision, including both repeatability and intermediate precision, to ensure consistency under various conditions. We also need to determine the limit of detection, or LOD, and limit of quantitation, or LOQ, which define the smallest amounts we can reliably detect and quantify. Robustness is another key point. It's about how well our method holds up under small, deliberate variations in parameters. System suitability tests ensure the entire system is functioning properly before sample analysis. When setting acceptance criteria for these parameters, we base them on stability data, product specifications, and the differences between release limits and shelf life limits. And of course, all these criteria must be scientifically justified to support our method validation process. By thoroughly understanding and applying these parameters, we help ensure our analytical methods are both reliable and compliant, supporting the overall quality and safety of our products. When it comes to regulatory expectations, it's essential to follow a science and risk-based approach. This means our methods and data should be grounded in scientific principles and aligned with the specific risks involved. Full method validation data is a must. This proves that our testing methods are reliable and reproducible. We also need to think about the lifecycle management of our methods, ensuring they remain valid and effective throughout their use. Data integrity and thorough documentation are critical. Regulators want to see clear, accurate, and unaltered records at every stage. Now, regarding the stability protocol design, we typically test a minimum of 3 batches. This helps us assess consistency and robustness across different production runs. Testing is done at various intervals, initially at 0 months, then at 3, 6, 9, 12 months, and so on to monitor stability over time. As for storage conditions, these are based on climatic zones, which range from I to IV. We generally test at 25 degrees C with 60% relative humidity, 30 degrees C with 65% RH, and 40 degrees C with 75% RH. This way, we can understand how the product behaves under different environmental stresses, ensuring safety and efficacy throughout its shelf life. All of these elements are interconnected. They help us meet regulatory standards while ensuring our products remain safe, effective, and of high quality for our patients and consumers. So, to wrap up, let's talk about why stability indicating methods are absolutely essential in our work. They play a critical role in ensuring drug quality and safety, giving us confidence that the product remains effective throughout its shelf life. They are also vital for regulatory approval, since authorities require solid data demonstrating product stability and consistency. It's important to remember that these methods must comply with key guidelines like ICH Q1, Q2, R2, and Q14. These standards help ensure our methods are scientifically sound and internationally accepted, which is crucial for global markets. Now, for success in this area, there are a few key factors to keep in mind. First, robust method development is essential. Our methods need to be precise, reliable, and sensitive enough to detect degradation products. Proper validation is equally important, confirming that our methods consistently produce accurate results. And finally, comprehensive degradation studies allow us to understand how our drug substance or product behaves under various stress conditions, helping us develop more stable formulations and ensuring safety and efficacy over time. In essence, these elements come together to support the overall quality and regulatory compliance of our pharmaceutical products. Thank you. For further support, please contact us. We're here to help you with any questions or additional information you might need. Don't hesitate to reach out. Your success is our priority. And with that, I want to thank you all for your time and attention today. It's been a pleasure sharing this information with you, and I look forward to connecting again soon.